Hello, my name is Jaycie Fickle, and I am a graduate student at California State University of Bakersfield. And the title of my project is Xylem Structure and Hydraulic Function in the Roots and Stems of Chaparral Shrub Species in the Sierra Nevada. First off, I'd like to acknowledge my faculty mentor, Dr. Anna Jacobson. I'd also like to acknowledge others who assisted with this research, Dr. Brandon Pratt, Marta Percola, Viridiana Castro, Aaron Baumgardner, Angela Madsen, and Jacob Spriester. I'd also like to acknowledge my CSUB CREST grant, which funded this project. So water transport in plants can be described by the cohesion tension theory. And in the cohesion tension theory, leaves are losing water to the atmosphere, and this creates a negative pressure gradient all the way down through the plant to the roots. And this creates a suction that pulls water in through the roots and up the plant body. Stems and roots are very different from each other. Stems are out in the open air, while roots are in the soil in contact with the water. In addition, stems are supported by cells in their plant body, while roots are supported by the soil. So xylem is responsible for water transport in plants. And within xylem are vessels. And these are the pipes through which water flows. And water can flow from one vessel to another through pits, which are these little holes in the sidewalls of the vessels. A major impact in water transport can be an embolism, or a gas-filled space. And this can occur in drought or in water stress conditions, where gases are pulled into the vessels, or this can occur in freeze-thaw stress, where during freezing, gases will come out of solution, and then during thawing, these gas bubbles will coalesce and form larger bubbles. And this is really important when considering water transport, because these emboli can block off water transport in the plant, and they can spread from one vessel to another, choking off large areas of transporting vessels in the plant. The size of vessels correlate to hydraulic function. So larger vessels are more efficient at moving larger quantities of water, but they are more vulnerable to these emboli occurring, while smaller vessels are more safe to these emboli, but they're not as efficient at moving as much quantities of water. So with that, I hypothesized that xylem traits will differ among the different organs of the plants because of their different functions and their different environments of their surroundings, so the soil versus the air. And with that, I predicted that roots would have larger pit membrane areas. So these holes in the side walls of the vessels would be larger in the roots than the stems. I also predicted that roots would have larger vessel diameters than the stems, and therefore that roots would be more vulnerable to cavitation in the stems, than the stems. So for this study, I did in the Lake Isabella Alta Sierra area, and for my thesis project, I'm looking at two different elevational gradients. But for the purposes of this presentation, I grouped the two sites together and only am looking at differences between stems and roots. And so I looked at 5 chaparral shrubs that occurred at these sites: Arctostaphylos biscuita, Cercocarpus fatuloides, Fremonodendron californicum, Garrya flavescens, and Quercus garryana. And so this summer I spent extracting roots from the soil using hand trowels. And once the roots were exposed, they would be clipped underwater. And this would be to prevent air bubbles from entering into the stems or the roots once they were clipped. And then the stems and the roots were transported back to lab in vials of water. And once I got them back to lab, I would trim them down to equal sizes underwater, and then I would flush them under high pressures to remove all air bubbles that were occurring within the stems and the roots. So for the hydraulic measures, I measured hydraulic specific conductivity, or KS, and then I did that using the hydraulic apparatus. So here is the hydraulic apparatus here, and so water is flowing through this tube, through the stem, and the output of water is being measured here on this scale. And so this measures the amount or quantity of water flowing through the stem or the root in a given time. And so I also measured the pressure at 50% loss in conductivity, or P50. And that was done using the centrifuge method. And so with that, I would measure the hydraulic conductivity, and then I would put it in the centrifuge, and the centrifuge simulates drought conditions. So it pulls on the water column within a stem or a root, and it creates these little emboli within them. And so then I would measure the hydraulic apparatus, I would measure the hydraulic conductivity again, and I would do this again and again at increasingly negative pressures. And I would do this until there was no flow left in the stem or the root. And from there, I could back calculate the pressure at which 50% of the conductivity was lost. And so for my results, I found that roots were less resistant to gas bubbles forming than the stems. So here in the white dots are the roots, and the black dots are the stems. And you can see here that the roots gave out conductivity a lot earlier than the stems did. Another interesting thing I found was that while the species differed from each other in hydraulic conductivity in P50 between species in the stems, those kinds of differences weren't seen in the roots. So all of the species were very similar to each other in the roots, but not in the stems. Some other things that I found were that roots were more efficient in their hydraulic characteristics and their xylem than the stems were. So these pictures down here are taken from a stem and a root of the same individual. So see, here you can see that the stems had much smaller vessel diameters than the roots did, and they also had larger pit membrane areas than the stems. So in conclusion, there were differences between species and their vulnerability to cavitation, especially in the stems. However, roots were similar between species considering their vulnerability to cavitation, and that there were large functional differences between the stems and roots within the same plant. So for my thesis, I plan to make some more comparisons. I plan to measure some more vessel diameters and their wall thickness. I plan to measure pit membrane areas, and I plan to measure the vessel length. And all of these traits correlate to hydraulic safety and efficiency and would help me explain some of the differences I saw between the stems and the roots. And thank you. And with that, I look forward to receiving any questions.